Title from Pathogens Pandemics commemorate the World Zoonosis Day. So you all must be knowing about viruses. What happens when virus that normally infects a non-human animal makes a jump in its host, starts to infect a human being. We call that event as zoonosis. Non-human virus infecting non-human animals who begin to infect. This has happened over several times. This was very, very gruesome. In the recent past, you all must have heard about swine flu, bird flu. Most recently, the major devastating pandemic that was COVID-19. So these are all examples of zoonosis. Viruses which was infecting some other animal suddenly started to infect humans because humans don't have immunological defense mechanisms to cope with these viruses. If we were not the original host of the virus. So it can be have a very devastating effect as can also pandemic. So we will hear about these various aspects of zoonosis from our resource persons. The first speaker we will be having Dr. Onivan Mitchell. So he used to be a microbiologist, now he's a science educator, teaching various courses and sciences, and very efficient and very nice science communicator. He writes popular science articles in various newspapers, magazines, and online blogs. He can really illustrate various complex mechanisms of science in a simple way. We will have his formal introduction later on. The second speaker we will be having Dr. Imran Banerjee, Associate Professor Aisha Mohali. He is actually a practicing biologist, research on virus. From him, you will hear about how we can, one of the ways by which we can prevent the entry of viruses in the cell. So that would be potential new therapeutic way of preventing pandemics. Before starting the program, we would like to have a small inauguration. Just on the principal ma'am, Dr. Malabos, and the speaker, Dr. Amanda Mitchell, all the faculty members who present here. Who will be the water of the class we have here. The incidences of zoonosis will keep on increasing in the recent future because of changes we are seeing in our environment, because of climate change, and because of the destruction of the human habitat, the destruction of the wild habitat that humans are causing. That can bring together viruses that we were not exposed to earlier to the humans. Very important to preserve our environment, not only for preventing climate change, reducing climate change in that aspects, but also for preventing zoonosis. Dr. Iswaran Miller is a person of Raleigh and Common States. Self-purpose speakers. Please put forward the question in the comment section. We will read out the questions to the speaker. Speaker will answer to you. Before we proceed with the house, first we would like to request our friends on my kindly speaker with the notes.
শুভ মধ্যাহ্ন ওয়ার্ল্ড জুনোসিস ডে উপলক্ষে সরকারি রাজ্য কলেজ বিমেনের জুলজি ডিপার্টমেন্ট আজকে এই সেমিনারের আয়োজন করেছে আমাদের অত্যন্ত গর্বের বিষয় কলেজে যে কটি ডিপার্টমেন্ট নিয়ে আমরা গৌরব করতে পারি তার মধ্যে অন্যতম হিস্টোলজি ডিপার্টমেন্ট। গবেষণার জন্যই হোক, আমাদের রিসোর্স যা আছে হিউম্যান রিসোর্স সেই জন্যই হোক, ইনফ্রাস্ট্রাকচারের জন্যই হোক, আমি বিভিন্ন কলেজে কাজ করেছি। কিন্তু আমি দেখেছি যে এখানে এই কলেজ যে একই এ গ্রেড পেয়েছে, প্র্যাকটিক্যালি সেটাও বোধহয় জন্মাত্রী উন্নয়ন নয়। আমাদের যা রিসোর্স আছে, আমরা ন্যাকের গ্রেডুয়েশন যাই থাকুক, আমাদের পাড়া প্রতিবেশী এবং যারা শিক্ষা জগতের সঙ্গে জড়িত আছে তাদের সবার বিচারে আমরা নিশ্চয়ই আরো অনেক এগিয়ে আছি। সামাজিক ভাবমূর্তি বলে একটা বিষয় হয়, এবং পরীক্ষার রেজাল্ট বলে একটা বিষয় হয়। যেমন অনেক সময় হয় কোনো ছাত্র যে মার্কসটা পেলো, পাড়ার সবাই বলে ও এটা পেলো, এর থেকে ও আরো অনেক ভালো। তো আমাদেরও সেই রেপুটেশন আছে সমাজে এবং শিক্ষা জগতে, যে এই কলেজটি ন্যাকে এ পেয়েছে না এ প্লাস পেয়েছে সেটা বড় কথা নয়, কলেজ সত্যিই গৌরব করার মতো জায়গায় আছে। এবং এই ধরনের অনুষ্ঠানগুলো সেই পরিচয়কে এবং সেই বিশ্বাসকে আরো বেশি দৃঢ় করে। আহ, আমি খুব আনন্দিত যে আজকে আমাদের মধ্যে উপস্থিত আহ, করছেন এবং আমাদেরকে সম্মিলিত করবার জন্য থাকছেন ডক্টর অনির্বাণ মিত্র এবং ডক্টর বিদুলাল ব্যানার্জি। উনি বললে পরের ধাপে আসবেন, ঘরের জন্য পরের ধাপে আসবেন। আমি স্বাগত জানিয়ে রাখছি। আমাদের মধ্যে উপস্থিত আছেন এই বিভাগের প্রাক্তন আমাদের অ্যাসোসিয়েট প্রফেসর ম্যাডামকে অশেষ ধন্যবাদ যে তিনি আহ, প্রাক্তন হয়ে যাবার পরেও আমাদের সঙ্গে সম্মিলিত হতে এসেছেন। আর যারা অতিথি হিসেবে এসেছেন তাদেরকে জানাই যে হয়তো আপনারা খবরে পাচ্ছেন যে আগামী দিনে হয়তো এই কলেজের জুলজি বিভাগ শুধুমাত্র ইউজি কোর্স করাবে না, পিজি কোর্স করানোরও সুযোগ পাবে। এটা আমাদের ছাত্রীদের জন্য খুব গৌরবের বিষয়। এবং আমি মনে করি যে বিশ্ববিদ্যালয় থেকে পুরনো যেহেতু এই কলেজ, আমরা এই বিশ্ববিদ্যালয়ের অধীনে আছি, তার থেকে অনেক বেশি পুরনো, 68 বছর হয়ে গেছে এই কলেজে। এবং সেই জায়গা থেকে অবশ্যই আমাদের ইনফ্রাস্ট্রাকচার অন্য অনেক জায়গা থেকে অনেক ভালো। কাজে পিজি যদি আমরা এই সেশন থেকেই চালু করতে পারি, সেই একটা খুব সম্ভাবনার জায়গা আমাদের সামনে তৈরি হয়েছে। তাহলে আমাদের ছাত্রীরা এবং আরো আমাদের জীবনের ছাত্রীরাও খুব উপকৃত হবে। আজকের অনুষ্ঠানে স্বাগত ঘোষণা করছি। ধন্যবাদ।
So, thank you. Now, I'd like to request Dr. Wandering on the kindly formally introduce the speaker.
Respected Principal Ma'am, IQSC Coordinator Ma'am, distinguished speaker, and my learned colleagues and my dear students. I have been bestowed with the honor and privilege to introduce today's one of the guest speaker, Dr. Anubhav Mitro, to this August gathering. Dr. Mitro graduated in physiology from Presidency College and did his master's in biotechnology from NS University, Baroda. He completed his doctoral studies on gene expression in bacterial system at the Department of Microbiology and Cell Biology, Indian Institute of Science, Bangalore. He has a good number of publications in international and national review journals. Subsequently, he became a teacher of molecular biology and biotechnology. Over the last few years, he has been conducting semesters in modern biology at Jadavpur University and PG section of Rammohan College under University of Dhaka. Dr. Mitra is also a passionate writer, an active blogger, and an excellent science communicator. His articles have been published by reputed journals, magazines, newspapers, both in English and Bengali. Today, he will deliver a lecture on the topic named tracking zoonosis and potential pandemics in the 21st century, which is very relevant for this day. It is an honor and privilege for us to listen to such an experienced academician. May I now request Dr. Mitro to kindly address the seminar.
Before we begin the talk of Dr. Lecture, like for semester students, and if I state a principal speaker. Grateful to the college, to the department for giving me this opportunity. And when Honorable suggested that I give this talk, I asked him that what's the main audience here? And he said it will be our students, the BSc students. So then I thought that I will try to tell you একটা গোয়েন্দা গল্প। Okay? I think going by আমাদের যা চলে in the market and in আমাদের বাংলা cinema, there's a lot of interest in detective stories. Too much interest at times it's like. So I will try to tell you about একটা medical detective story. Uh, I presume everybody is comfortable with both English and Bengali, so I will keep on shifting between that. And I can see two of my students here. Oh, uh, so what we will be discussing is how HIV became the human pathogen. So HIV, which causes the disease AIDS, how did it become the human pathogen? Now, first thing we have to remember here, so there are two things that you should remember. First of all the diseases that we know till today, so TB, cholera, typhoid, smallpox, dengue, everything, okay, they are caused by natural pathogens. Some virus, bacteria, protozoa, something which has come from nature. That is rule number one. Second point, how did they infect humans? Most of these pathogens have come from some animal into the human society. Okay, some have come 10,000 years back, some have come recently, but they have, most of them have come from some animals. That means they are zoonoses. Alright? Now, our story starts with 1981 when first in the United States doctors noticed that there was a rare cancers and lots of opportunistic infection. Okay. Second. Right up, যে in 1981 দেখা গেল যে আমেরিকাতে many people were turning up যাদের lots of opportunistic infections হচ্ছে, অর্থাৎ যেসব ইনফেকশনস নর্মালি লোকজনের হয় না সেসব হচ্ছে, and very rare cancers হচ্ছে, and the first people who were getting these infections were mainly homosexual people. And then it was seen that the main জিনিসটা যেটা হচ্ছে যে এদের ব্লাডে টি হেল্পার লিম্ফোসাইটস প্রচন্ড কমে গেছে। And then it was realized it was not only homosexual individuals, it was also with all kinds of people, men, women, even যারা কোনো একটা infected syringe থেকে নিচ্ছে, infants from their mothers, everybody was getting it. Okay. So it was then realized that actually either it was a virus, the pathogen যেটা blood থেকে আসছে, কিংবা some kind of body fluid, some sexual fluid থেকে আসছে। Now, over time, this disease has spread across the world and it has become a pandemic যেটা কিন্তু এখন চলছে। This is AIDS, acquired immune deficiency syndrome. যখন ভাইরাসটা বডির ইমিউন সিস্টেমকে ডিস্ট্রয় করে দেয়, তখন as the T helper lymphocytes die, all kinds of infections and cancers start taking over. So the patient dies after few years. Alright. So this is how the infection started spreading. 1981. 2 years later, Luke Montagnier, Robert Gallo, they identified যে ভাইরাসটা এই অসুখটা করাচ্ছে, and it was named HIV-1, human immunodeficiency virus. Now the thing যেটা মনে রাখার সেটা হচ্ছে this is একটা retrovirus. এই ধরনের বাল্টিমোর ক্লাসিফিকেশনে there are 7 kinds of virus. তোমরা জানো যে আমাদের যে হিউম্যান জিনোম that is double stranded DNA, right, chromosome cells. Now ভাইরাসের জিনোম can be RNA or DNA, double stranded, single stranded সব হতে পারে। Among them এই retrovirus যাদের মধ্যে HIV টা পড়ে, they have single stranded RNA genome যেটা during infection একটা double stranded stage আসে। এটা আমাদের for our present talk it's not that important, but এই জিনিসটা মনে রাখতে হবে that these kind of retroviruses can infect many mammals. Okay, এবং রবার্ট গ্যালো, লুক মন্টাগনিয়ার এটাকে আবিষ্কার করেন। Question is, HIV এলো কোত্থেকে? From which animal did it come from? That is the question. Okay. Now, দেখো when we are studying যে একটা organismal evolutionary history, what we do? We go for fossils, we go for comparing the other anatomical, physiological properties, and then আমরা একটা phylogenetic tree, যে এই মানুষ তার সঙ্গে শিম্পাঞ্জির এই রিলেশনশিপ, আর দূরে গরিলা, ওরাংওটাং, ওখানটাই। So we can construct একটা phylogenetic tree, right? Today, when we do that, we also use the molecular clock. Now, molecular clock ব্যাপারটা কি? Molecular clock ব্যাপারটা, I'm talking on this because it will come back in our talk again, in the topic again. Let's say take the example there are two genomes, human DNA and orangutan DNA. Fossil evidence থেকে we know that they separated 30 million years ago. এইবারে if you compare human DNA and orangutan DNA, তুমি দেখবে human DNA তে যেখানে A আছে, orangutan DNA তে C আছে। Alright, so there are certain changes. Now তুমি compare করলে, what did you see? Orangutan DNA আর human DNA এর মধ্যে differences আছে, let's say 40. আর how many years since they became separate? 13 million years. তাহলে 40 by 13 total differences কত? 3 changes per million years. ঠিক আছে, 40 by 13 approx. এই যে তুমি একটা রেট পেলে, এটা হচ্ছে মলিকুলার ক্লকটাকে তুমি ক্যালিব্রেট। You know there are 3 changes in DNA sequence per million years. এবার ধরো তুমি একটা অন্য অ্যানিমাল দেখছো, let's say তুমি looking at pig. Okay, pig DNA, human DNA. You compare. Make this an example. Just you'll see that the difference হচ্ছে, let's say the difference যেটা এখানে ছিল 40 between human DNA and pig DNA, it is 150 differences. তাহলে তুমি আগের হিসাবটা ধরে what can you say, 150 by 3, 50 million years এর difference. তাহলে এইরকম করে you can construct phylogenetic trees. You can find out তোমার কাছে যদি DNA sequence থাকে বা RNA sequence থাকে, you can compare them, and যত বেশি difference তত সেই organisms were far apart. যত close তত they were the same organism, তারপর diverge করে আলাদা species হয়ে গেছে. Am I clear? Now, এইটা becomes a big advantage for viruses. Why? Virus এর তো কোনো fossil হয় না. So if you have to do analysis, you have to look into this kind of sequences. Second, viruses, they multiply very fast. Plus, they have mutations much faster than organisms like humans. What do I mean by that? Let's say, এখন 2024, 1920 তে একটা ভাইরাস থেকে দুটো আলাদা স্পিসিস হয়ে গেল। In 100 years, 50টা কেন, ওর দুজনের মধ্যে 1000 differences হয়ে গেছে in only 100 years. তাহলে তুমি এবার ক্যালকুলেট করতে পারো per year 10 differences হয়েছে. Accordingly you can calculate কত কত ভাইরাসটা চেঞ্জ করতে করতে যাচ্ছে. এই যে let's take the example of COVID. যখন COVID প্রথম এলো, we had the alpha strains, then we had the delta strain, then omicron strain. All these strains change করতে করতে যাচ্ছে, right? So you can use this kind of information to find out which virus came earlier, which came later, তাদের মধ্যে relationships কি, all of this you can track. ঠিক আছে? Now, going back to the AIDS. 1981 এ আমেরিকাতে দেখা যাচ্ছে patients have AIDS. ঠিক আছে? Same time দেখা যাচ্ছে আমেরিকাতে প্রাইমেট রিসার্চ সেন্টারসে ক্যাপটিভ যেসব ম্যাকাক্স, এই রিসার্চ মাঙ্কি ধরনের যেসব ম্যাকাক্স আছে, তাদেরও সিমিলার একটা ডিজিজ হচ্ছে। ঠিক আছে? and many of these monkeys were dying. ঠিক আছে? এই অসুখটার নাম দেওয়া হলো সিমিয়ান এইডস। ওকে, তখন এখানকার প্রাইমেট রিসার্চ সেন্টারে যে সায়েন্টিস্টরা, they started looking back ওদের যেসব পুরনো রেকর্ডস, they found that not only in 1981, 1970 এরকম অসুখ হয়েছিল কিছু কিছু, আরো they looked in detail, they found that যতগুলো macaques got this disease, they had all come from California Primate Center. এই অসুখটা লেট 1960s থেকে মাঝে মাঝেই হচ্ছে। তার মানে দেখো, this was happening some 14-15 years before human AIDS started. আরো করতে করতে তাহলে কি করতে হয় এবার? Soon they identified the virus যেটা এই বালকদের মধ্যে আছে, সেটার নাম দেওয়া হলো সিমিয়ান ইমিউন ডেফিসিয়েন্সি ভাইরাস মেকা। ঠিক আছে, দেখা গেল this is the virus that causes the disease in the monkeys. Okay, after some time দেখা গেল আরো monkeys আছে, এই stump tail macaque, সেখানেও একটা SIV পাওয়া। ঠিক আছে, তাহলে as time goes on, more and more simian immunodeficiency virus, সেটা হলো SIV, these were discovered. এখন প্রায় 40টা আছে। অলরাইট, এইবারে প্রশ্ন হচ্ছে, একটা কনফিউশন স্টার্ট। What is the confusion? ভাইরাসটা অরিজিনালি where did it come from? এটা কি এই monkeys থেকেই এসেছিল or some other species? What is the problem? The problem is, যখন এইসব বাদরগুলো when they are kept in the primate centers, এগুলো আলাদা আলাদা খাঁচায় থাকে, they stay in large open areas like this, যেগুলো বলে corals, এই open areas এ থাকে, 50-100 monkeys stay together. এবার যখন থাকে, then মাঝে মধ্যেই মারপিট হলে কামড়াকামড়ি আঁচড়াচড়ি হয়। So now the question comes, যে it is possible these monkeys got the virus from some other species, করে ওদের অসুখটা হয়েছে। তাহলে question is, which species was the primary source of this
SIV virus? That became the question. All right. এই করতে গিয়ে to find out this answer, scientists started looking in detail for SIVs. যেখানে যা SIV আছে সব they started looking in. এবং what turned out is more than 40 SIVs are found only in wild African old world monkeys, but nothing in Asian monkeys. Alright? তার মানে বোঝা গেল যে ওই মাকাকগুলো তো এশিয়ান, but they had the virus from some African monkey যেটা ওদের সঙ্গে একই এরিয়াতে ছিল in those research. তাহলে সেটা খোঁজা হলো, and what they discovered was that একটা মাঙ্কি আছে ওয়েস্ট আফ্রিকান সুটি ম্যাঙ্গাবে মাঙ্কি, ওকে, সেইটার মধ্যে যে ভাইরাসটা পাওয়া গেল এই এসআইভি এসএমএম, এইটা হচ্ছে এই ভাইরাস গুলোর অরিজিনাল সোর্স। তাহলে কি বেরোলো ফাইনাল এসআইভি এসএমএম from this African sooty mangabe monkey. This was the original virus. These monkeys were staying in the primate research centers with the Asian monkeys. এবং সেখানে during some fight ভাইরাসটা এই স্পিসিস থেকে ওই স্পিসিসে ট্রান্সমিটেড হয়ে গেছে, পরে তারপর ইভলভ করে নতুন ভাইরাস তৈরি হয়েছে। সো এই জিনিসটা তখন এখান থেকে গট ডিসকভার। আরো ডিটেলস হুইচ সেন্টার, তখন দেখলো আমেরিকাতে এইরকম সুটি ম্যাঙ্গাবে মাঙ্কিস থাকতো 4টে সেন্টারে, 3টে লুজিয়ানায়, একটা ক্যালিফোর্নিয়া। Now scientists started doing detailed ওই phylogenetic molecular block analysis, যত ভাইরাস আছে বার করে তাদের সিকোয়েন্স দেখছে। When they did all this analysis, what they found was যে 1920 থেকে 1968 এই সময়টা ওয়েস্ট আফ্রিকা থেকে আমেরিকাতে সুটি ম্যাঙ্গাবে মাঙ্কিস এক্সপোর্ট হতো। ওকে, ওয়াইল্ড মাঙ্কিস আফ্রিকাতে ধরা হতো, দে উড বি ট্রান্সপোর্টেড টু ইউএস। 9 of these monkeys যখন তাদের এক্সপোর্ট করা হয়, দে হ্যাড দ্য ভাইরাস অলরেডি, দে ওয়্যার ক্যারিং দ্য ভাইরাস অলরেডি। They went to USA and then they spread the virus to the other monkeys in the cells. ঠিক আছে, তাহলে এইবার বেরিয়ে গেল যে twice at ক্যালিফোর্নিয়া সেন্টারে দুবার এই এসআইভি এসএমএম আফ্রিকান মাঙ্কিস থেকে এশিয়ান ম্যাকাকসে ছড়িয়ে গেছে, এবং তারপর যেই ছড়িয়ে গেছে নতুন ভাইরাস তৈরি হয়েছে এবং ম্যাকাকসের মধ্যে সেক্সুয়াল কন্ট্যাক্ট, মাদার চাইল্ড ট্রান্সফার, ইনজুরি, এরকম করে করে they are spreading. Now what I want you to remember is that this is a West African monkey virus যেটা স্প্রেড করে গেছে, এটা রিমেম্বার। We go back to human AIDS. সেটা হচ্ছে 1979 এ পিটার পায়ো, who was a doctor scientist in Belgium, he was getting two kinds of patients. One were people যারা আফ্রিকা থেকে এসছে, মানে আফ্রিকান অরিজিন, যারা বেলজিয়ামে এসছে ট্রিটমেন্ট করাতে। সেকেন্ড হচ্ছে পিপল হু আর ইউরোপিয়ান, কোনো কারণে আফ্রিকায় গেছে। They are getting all these unusual infections and cancers. Alright, ছেলে মেয়ে সবাই আছে। Two, he was also getting patients, mainly homosexual men, যারা who had traveled to the USA থেকে ফিরেছে after few months, 1 year, 2 years, they are coming to him because of similar diseases. Now Peter Payo connected the two. একদিকে আফ্রিকা থেকে লোকজন আসছে এই ধরনের সিমটমস নিয়ে, সিমিলার সিমটমস নিয়ে আমেরিকা থেকে ফেরত সাবটিকুলার কাম। Peter Payo realized that this is the same disease. Both sets of people are getting the same disease, similar symptoms. Men, women, homosexual, heterosexual, everybody is getting it. It is a sexually transmitted virus. এবং সেম টাইম ভাইরাসটা আবিষ্কার হয়ে গেল HIV-1. So he realized that this is the same pathogen. Second হচ্ছে, তুমি ভেবে দেখো, how many people can come from Africa to Belgium for treatment? It's very costly. Not many people. That means maximum লোক আফ্রিকাতে আছে, they are actually sick, they cannot come to Belgium. So if you go to Africa, you will get many more patients. So Pio traveled with scientists to Kinshasa, যেটা হচ্ছে Democratic Republic of Congo তে, এবং সেখানে it was shocking. Entire hospitals full of terminally sick patients. All right. So massive AIDS epidemic across Central Africa. And by 1987, it has spread to 37 countries: Democratic Republic of Congo, Rwanda, Burundi, Uganda, Zambia, Tanzania. But it's just at the massive epidemic spreading. No way to stop it. All right. Same time, another information came. Central Africa, HIV-1 spreading like wildfire. Western Africa, দেখা যাচ্ছে যেখানে ধরো এই সেনেগাল, গাম্বিয়া, গিনি বিসু এইসব সিয়েরা লিওন, লাইবেরিয়া এসব জায়গায় এখানে কিন্তু very few AIDS patients. Why? Strange, পাশেই তো এত হচ্ছে. Why here so less? So people started investigating এবং এই সেনেগালে যেসব সেক্স ওয়ার্কার্স সেখানে they started checking them for HIV তাদের ইনফেকশন হয়েছে. What they found was very surprising. 7% people are HIV positive. তার মানে তাদের ব্লাডে anti-HIV antibody আছে. All right, but the virus is not HIV-1, যেটা has been discovered earlier. It is একটা নতুন ভাইরাস যেটা কয়েকদিন পরে কন্টাক্ট নিয়ে আবার আবিষ্কার করে ফেললেন. It is HIV-2. এবং why is this special? Because HIV-2 is very similar to the simian immunodeficiency virus যেটা ম্যাকাক্সের আবিষ্কার. যখন সিকোয়েন্সটা কম্পেয়ার করলো তখন দেখা গেল the HIV-2 is very similar to the virus which had been found in the monkeys. এইবার তাহলে কথা হচ্ছে, where did HIV-2 come from? High chance from some monkey in Africa. Now when you আরো details analysis করলো, if you remember কয়েকটা slide আগে I told you, where did the Asian monkeys get the virus? From the African sooty banded monkeys. Compare করে দেখলো HIV-2 sequence আর sooty banded monkeys the sequence ভীষণ মিলে যাচ্ছে. So the original source of the HIV-2 is this virus যেটা ছিল sooty banded monkeys. This কি করে এলো? তাহলে যেমন it had spread from African monkeys to Asian monkeys in USA. In West Africa, দেখো West Africa তে এই এরিয়াতে sooty banded monkeys টা পাওয়া যায়. আর exactly এই এরিয়াতেই you were having HIV-2 maximum pollution. So this is another example of cross-species transmission. Sooty banded monkeys coexist with humans in West Africa. এবং এইখানেই must have happened monkeys থেকে humans এ ভাইরাসটা জাম্প করে গেছে. All right. How did it happen? This happened because of যাকে বলা হয় bushmeat trade. Bushmeat trade-টা কি? Bushmeat হচ্ছে meat of wild animals killed in the forests and savanna of Africa. দেখো চিরকালই আফ্রিকাতে for food purpose animals are hunted. The trouble is last 100 বছরে this has become huge. This has become so huge commercialized that per year 6 million tons of bushmeat, all kinds of small and large mammals, monkeys, apes, everything small, even elephants, everything is hunted for food and sold in the markets. All right. So this is a major problem. You can see from various sources have taken this picture. And এটা is a massive threat to wildlife. And এটার জন্য the major chances যত এরকম হবে, the chances of viruses from other animals entering humans is increasing. যাকে বলে cross species transmission. This is a very high chance happening over here. You can see pictures of bushmeat trade. There are chimpanzees and other animals, monkeys, everything has been killed for bushmeat trade. and you can see যখন এইরকম হান্টিং হচ্ছে, যখন বুচারিং হচ্ছে, তখন there is a high chance of some kind of injury, কোনো একটা কামড় দিল, ছুরি দিয়ে হাত দিতে গেল, সামথিং অর দি আদার, তার ব্লাড থেকে and in humans এ ঢুকে আসছে না। This is must be the case যখন ওই সুটি ম্যাঙ্গাবে monkeys এর infected blood থেকে humans in West Africa got the virus এবং over last 50-70 years সেটা HIV-2 তে evolve করেছে। তাহলে what got established that HIV-2 originated from সুটি ম্যাঙ্গাবে monkeys when এখানে আবার খুব ইন্টারেস্টিং একটা জিনিস, যখন ওইসব ফাইলোজেনেটিক ট্রি মলিকুলার ক্লক করা হলো, দেখা গেল this mainly happened around 1945-50 before 1960। এবার 1960 অবধি the ভাইরাসটা আছে, it's there in Africa, HIV-2, কিন্তু খুব কম। তারপরে it starts heavy infection, epidemic শুরু হয়ে যাচ্ছে। দুটো প্রশ্ন, why and where did the epidemic start and why did it start? Two questions. What are the answers? Best answer possible. There are a ছোট্ট দেশ আছে Guinea-Bissau, all right? A Guinea-Bissau is important to us এখন because এই দেশটাতে you get maximum HIV-2 cases, HIV-1 বরং কম, HIV-2 অনেক বেশি। তার মানে কি, যেখানে সবচেয়ে বেশি কেস সেটাই এপিসেন্টার হওয়ার হাইয়েস্ট চান্স। So a Guinea-Bissau is the epicenter of HIV-2 epidemic. All right. Next হচ্ছে, it started spreading from 1960s. Guinea-Bissau তে 1960s এ একটা জিনিস হয়েছিল, সেটা কি, যেমন আমরা ছিলাম ব্রিটিশদের আন্ডারে, Guinea-Bissau was a পর্তুগিজ কলোনি, মানে যেমন গোয়া পর্তুগালের আন্ডারে ছিল, তাহলে এই অ্যাঙ্গোলা, মোজাম্বিক, গিনি-বিসাউ এগুলো পর্তুগিজদের আন্ডারে ছিল। 1963 to 1974 আমি উইকিপিডিয়া ছবিটা লাগিয়ে দিয়েছি, গিনি-বিসাউতে একটা ফ্রিডম মুভমেন্ট শুরু হয় and it was like একটা বিশাল লড়াই। So Portugal sent thousands of their soldiers, এখানকার লোকজন লড়াই করছে, populations are being mixed, হ্যাঁ, বহু লোক মারা যাচ্ছে, all kinds of things are happening। ঠিক সেই সময়টাতে the virus starts multiplying, তাহলে একটা বিশাল সামাজিক চেঞ্জ, all kinds of thousands of people are moving around, hospitale injured, অনেক ধরনের খাবার দাবার নতুন আসছে, খাওয়া হচ্ছে, অ্যানিমেল ট্রেড চলছে, এভরিথিং ইজ হ্যাপেনিং, এবং HIV-2 এই অবস্থায় start spreading। তার মানে what all we can see, it was an African monkey that was the main source, cross species transmission to humans, where সব আনসার পাওয়া হয়ে গেল for HIV-2, all right? আচ্ছা, we go back to HIV-1, যেটা হচ্ছে আমাদের প্যান্ডেমিক ভাইরাস, যেটা অ্যাক্রস দ্য ওয়ার্ল্ড আছে। দেখো মনে করে দেখো, HIV-2 ওই ওয়েস্ট আফ্রিকাতে, HIV-1 is spread all over the world, including millions in India, all right? So HIV-1 করতে গেলে, all right, so অলরেডি জানা ছিল 1960-70s এ হয়তো কিছু কিছু কেস হয়েছে, বাট nobody knew। এইবার now what scientists started, they started to go back to very old records, মানে পুরনো হসপিটালের রেকর্ড, পুরনো কোথায় কে কবে আলমারির মধ্যে ব্লাড স্লাইডস রেখে দিয়েছে, কোথায় কোন টিস্যু আছে রেখে দিয়েছে, they began to find out সেখান থেকে HIV RNA পাওয়া যায় কিনা। ভেরি ডিফিকাল্ট জব, ওকে, খুব ডিটেইল কাজ। সে 50 বছরের পুরনো স্লাইড বার করেছে, সেখান থেকে রক্ত থেকে দেখছে HIV RNA পায়, ইটস লাইক দ্যাট। করে two things were found, two people were found। একটা ছিল একজন নরওয়ের একজন সেলার, এই নরওয়ে নেভিতে কাজ করতো, he had traveled to West Africa, he returned home in 1966, তারপর থেকে he started falling sick, after some time his wife also fell sick, both of them died, their youngest child also died। এবার এদের স্যাম্পলস, এদের টিস্যু স্যাম্পলস হসপিটালে স্টোর করা ছিল। When those samples were checked, it was found all three were HIV positive। Number one. Number two, sheer good luck that আমেরিকার একটা ইউনিভার্সিটিতে অনেক পুরনো আফ্রিকান ব্লাড স্যাম্পল স্টোর করে রাখা ছিল। ওকে, these blood samples had been collected in 1959 mainly, প্রায় 800 স্যাম্পল রাখা ছিল। নাও সায়েন্টিস্ট সেই পুরনো ব্লাড স্যাম্পল বার করে দেয়ার টেস্টিং, এখানে কোথাও এইচআইভি আরএনএ আছে, এইচআইভি অ্যান্টিবডি আছে, চেক করতে 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 800-র মধ্যে দে ফাউন্ড ওয়ান, একজন অ্যাডাল্ট ব্যান্টু মেল আফ্রিকান, তার কোড নেম L70, তার নাম কেউ জানে না। ইম্পর্টেন্ট থিং, হি লিভ ইন লিওপোল্ডভিল ইন বেলজিয়াম কঙ্গো, যেটা আজকের কিনশাসা বলে শহর, একদম সেন্ট্রাল আফ্রিকার ম্যাপে দেখবে বড় শহর কিনশাসা, সেইখানকার হি ওয়াজ এ রেসিডেন্ট। তার এইচআইভি 1 ছিল কবে? 1959-এ। অলরাইট, তার মানে এইচআইভি 1 was present in urban central Africa since 1959, 25 years। 84 সালে আইডেন্টিফাই হচ্ছে ভাইরাসটা, আসলে 1959-এও ছিল। যখন ছিল নিশ্চয়ই সিটিতে একজনের ছিল না, অন্য অনেকের ছিল, রাইট? বাই দিস টাইম আরো কিরকম রেজাল্টস দেখো, এইচআইভি নিয়ে রিসার্চ চলছে অ্যাক্রস দ্য ওয়ার্ল্ড, সায়েন্টিস্ট আর ফাইন্ডিং সিকোয়েন্স, সেগুলো বার করছে, সেগুলো স্টাডি করছে, করতে করতে দেখছে এইচআইভি সিকোয়েন্স সেগুলো থেকে আরো ডাইভার্স হতে হতে একটা আছে গ্রুপ এম, এবং তার থেকে আরো সাবটাইপস। দিস আর ভাইরাসেস যেগুলো পেশেন্টস তে পাওয়া যায় অ্যাক্রস দ্য ওয়ার্ল্ড, ঠিক আছে? সো দেয়ার আর সাবটাইপ এ বি সি সব পাওয়া যাচ্ছে। There are some recombinant forms virus, দুটো ভাইরাস যখন একই সেলে থাকে তাদের মধ্যে একটা হাইব্রিড তৈরি হয়, তাকে বলে রিকম্বিনেশন। সো এইরকম প্রচুর ভাইরাস পাওয়া যায়। স্যার এগুলো কি বলা হলো? গ্রুপ এম, মেইন গ্রুপ, মেজর গ্রুপ। তারপর কিছুদিন পর দেখা গেল 1990-তে গ্রুপ ও আবিষ্কার হলো। কোথায় আবিষ্কার হলো দেখো? ক্যামেরুন, সেন্ট্রাল আফ্রিকা। তারপর গ্রুপ এন পাওয়া গেল কোথায়? ক্যামেরুন। তারপর গ্রুপ ও, আবার ক্যামেরুন। তাহলে বারবার আমরা কি দেখছি বলতো? সেন্ট্রাল আফ্রিকা ঘুরে ফিরে আসছে। অল এইচআইভি ভাইরাসেস ম্যাক্সিমাম ফোকাস কবে? 1959, গ্রুপ এম কোথায়? সেন্ট্রাল আফ্রিকা। এগুলো এন ও পি কোথায়? সেন্ট্রাল আফ্রিকা, ক্যামেরুন। অলরাইট, এই করতে করতে যখন ওই L70 যে ওই 1959-এর যে কেসটা ছিল তার আরএনএ যখন অ্যানালিসিস করা হলো, তখন দেখা গেল এইটা হচ্ছে তোমার একটা টিপিক্যাল একটা ফাইলোজেনেটিক ট্রি এরকম দেখানো হয়। এইখানে হচ্ছে বলতে পারো অরিজিন অফ দ্য এইচআইভি 1 ভাইরাস। দেখো এই যে অ্যারোটা, দ্য রেড অ্যারো, দ্যাট ইজ দ্য আরএনএ অফ দ্য 1959-এর ওই আফ্রিকান লোক, ওর শরীরে যে ভাইরাল আরএনএটা পাওয়া। হোয়াট ডাজ দিস টেল আস? একদম যখন ভাইরাসটা ভেরি আর্লি ইন দ্য ভাইরাল হিস্ট্রি, you had this patient was a infected. Where did the patient live? কিনশাসা, সেন্ট্রাল আফ্রিকা। ঠিক আছে, তাহলে এই সব রকম ডেটা দেখে conclusion, all known subtypes and most CRF are co-circulating in and around Kinshasa. Conclusion, HIV-1 has probably been circulating in Kinshasa for longer than anywhere else in the world, and Kinshasa is the epicenter of the HIV pandemic. বারবার যেখানে ভাইরাসটা সবচেয়ে পুরনো, সবচেয়ে বেশি, all the diversity পাওয়া যায়, that must be the source. All right, why and where, why and when in Kinshasa? So here comes this scientist Michael Borobi, a very famous virologist. Michael Borobi আরো সব পুরনো HIV samples পেলেন, and he did again further analysis, and what he found was, যখন এইসব phylogenetic tree করলেন, molecular clock analysis করলেন, what he found was that HIV group F, যেটা মেইন যেটা প্যান্ডেমিক ভাইরাস, সেটা originated in Kinshasa around 1884 and 1924 একদম এই 40 বছরের মধ্যে একদম টাইম স্প্যানটা বলে দিচ্ছি তাহলে কোশ্চেন আসছে why in 1884 why in this time period here comes আবার দেখো এখানে you find আফ্রিকান যে হিস্ট্রি সেটা হচ্ছে if you see আফ্রিকার ম্যাপটা আমরা যেমন we were under British rule you can see আফ্রিকার কতটা এরিয়া was under British rule French rule এবং এই মাঝখানটা দেখো এটা ছিল আন্ডার বেলজিয়ান অলরাইট তো বেলজিয়ামের তখন রাজা ছিল লিওপোল্ড দ্য সেকেন্ড এই জায়গাটার নাম ছিল কঙ্গো নদী আছে বিশাল কঙ্গো ফ্রি স্টেট এই এরিয়াটা রুলার ছিল এবং হি ওয়াজ এক্সট্রিমলি ক্রুয়েল মানে ব্রিটিশরা ওর কাছে ছেলে মানুষ হি ওয়াজ এক্সট্রিমলি ক্রুয়েল এবং টু থিংস হ্যাপেন এখান থেকে বড় বড় শহর তৈরি করা হলো এবং মানে কঙ্গোর যা যা জিনিস আছে রিসোর্সেস আছে সব বেলজিয়ামে চলে যায় টু থিংস হ্যাপেন টু ডু দিস তাহলে বড় শহর লাগছে বড় পোর্ট লাগছে তাহলে দিস ইজ দ্য টাইম যখন কিনশাসা শহরটা ওয়াজ এস্টাবলিশড তাহলে আমরা কি কি পাচ্ছি উই আর ফাইন্ডিং যে 1890s নাগাদ কিনশাসা শহরগুলো ওই বেলজিয়াম কঙ্গোতে এস্টাবলিশ হচ্ছে সেখানে থাউজেন্ডস অফ পিপল আর কামিং টু ওয়ার্ক জাস্ট থিংক অ্যাবাউট আমাদের মানে ক্যালকাটা বম্বে মাদ্রাস অল দিস ওয়্যার এস্টাবলিশড ফর কমার্শিয়াল ইন্টারেস্টস অফ দ্য ব্রিটিশ একই জিনিস ওখানেও তাই তাহলে থাউজেন্ডস অফ লোকজন কাজ করতে আসছে প্রচন্ড ভিড় তারা গাদাগাদি করে থাকছে সেকেন্ড বেলজিয়ান পলিসি হচ্ছে শহরে কাজ করতে অনলি দ্য মেলস উইল কাম মেন উইল কাম মহিলারা সব গ্রামে গঞ্জে থাকে সো দেয়ার ইজ দিস হিউজ ইমব্যালেন্স ইন দ্য সিটি জেন্ডার ইমব্যালেন্স হোয়াট হ্যাপেন্স এরকম হলে বিশাল সেক্স ট্রেড শুরু হয়ে যাচ্ছে and that is where HIV কিন্তু কাছাকাছি ছিল HIV-1 কবে হয়েছে 1884 1924 আর কিনশাসা শহরগুলো কবে হয়েছে 1900 প্রচন্ড ভিড় শহর HIV সেখানে স্প্রেড করতে হবে not only HIV all STDs started to spread এবং HIV also started to spread so by 1960 কিনশাসার পপুলেশন হয়ে গেছে ছিল 16,000 হয়ে গেছে 4 লাখ extremely crowded cities এবং this is where HIV epidemic starts ঠিক আছে এই হলো HIV so Leopoldville and Kinshasa is the best place for survival and later expansion of HIV-1 এবং ওর যে পেপার এই বরবির যে পেপার নেচারে সেখানে বরবি অ্যাকচুয়ালি বলছেন the rise of cities may have facilitated the establishment and early spread of HIV-1 hence the founding and growth of colonial administrative and trading sectors such as Kinshasa তার মানে একটা ভাইরাস ছিল এমনি রুরাল এরিয়াতে ঘুরছিল খুব বেশি লোকের ইনফেকশন হচ্ছিল না যেখানে ম্যাক্সিমাম লোকজনের ভিড় হলো কোথায় হলো শহরে হলো সেখান থেকে ভাইরাসটা বেশি বেশি করে ছড়াতে হবে সেই লোকজন আবার তাদের গ্রামে ফিরে যাচ্ছে তার অন্য কোথাও যাচ্ছে the virus is spreading more which animal was the source of this virus আমাদের কাছে আগে থেকেই জানা আছে যে আফ্রিকান monkeys থেকে ভাইরাস HIV-2 ছড়িয়েছিল তো সায়েন্টিস্ট started looking for other SIVs এবং অন্যান্য SIV দেখো ব্যাঙ্গল থেকে পাওয়া গেল আফ্রিকান গ্রিন মাঙ্কিস থেকে পাওয়া গেল বাট এগুলো সব মানে ফেলিওর দেখা গেল HIV এদের সঙ্গে অতটা রিলেটেড নয় দেন these scientists মার্টিন পিটার্স এলিক ডেলাপোর্টে এরা রিসার্চ করছিলেন শিম্পাঞ্জিদের they were checking the blood of chimpanzee and when they checked chimpanzee blood they luckily found যে দুটো শিম্পাঞ্জি আছে ওদের পেয়েছেন মানে তারা চিড়িয়াখানায় বা প্রাইভেট রিসার্চ সেন্টারে থাকে যাদের ব্লাডে there is a virus SIVCPZ যেটা HIV-1 এর সঙ্গে খুব ম্যাচ করছে সিকোয়েন্স যখন কম্পেয়ার করছে the chimp virus and HIV-1 very close match করছে। অলরাইট, some days later বেলজিয়ামে একটা চিড়িয়াখানাতে একটা শিম্পাঞ্জি ছিল, তার ব্লাড থেকে পেয়ে গেল আর একটা এইচআইভি, এরকম ভেরি সিমিলার ম্যাচিং উইথ এইচআইভি 1। এবং not only in Europe, Beatrice Hahn was working in the United States. She was also checking for the blood of chimpanzees. She also found this. This chimpanzee, jar blood there is a virus which very closely resembles HIV-1 virus. The chimpanzee virus সঙ্গে human HIV-1 virus very closely matching. All right. So all this was found out. এবং সেখানে this person came, Paul Sharp, is a very famous scientist. He started looking into details of all this sequence analysis. কি বেরোলো তার আগে একটা কথা শিম্পাঞ্জি নিয়ে, I think all of you know better than me, because the chimpanzees are broadly two types. Chimpanzees are the common chimpanzee and the bonobo chimpanzee, the common chimpanzee and the bonobo chimpanzee, and they all live in equatorial Africa. Bonobo chimpanzees live in this green area south of the Congo River, and the common chimpanzee তার আবার চারটে সাবস্পিসিস আছে, তাহলে ইস্টার্ন, সেন্ট্রাল, নাইজেরিয়া, ক্যামেরুনিয়ান এবং ওয়েস্ট। They all live in this park. All right, তাহলে আবার সেই সেন্ট্রাল আফ্রিকা ফিরে এসেছে, isn't it? এইবার যেটা শুরু হলো, অলরেডি ইউরোপ এন্ড আমেরিকাতে কিছু চিম্পাঞ্জির ব্লাড ভাইরাস পাওয়া গেছে which is matching HIV-1. মনে রাখবেন, ঠিক আছে? ওকে, so let us go for what happens in the forests of Africa. So the scientists now travel to Tanzania, যেখানে বোম্বে ন্যাশনাল পার্ক, এবং সেখানে ওয়াইল্ড চিম্পাঞ্জিদের স্টাডি করতে আরম্ভ করলেন যে এদের মধ্যে কি এই ভাইরাস পাওয়া যাচ্ছে। অলরাইট, সো দিস ইজ নট ইজি, রাইট? নিশ্চয়ই বনের মধ্যে গিয়ে চিম্পাঞ্জিদের প্যাক করে একটা ইনজেকশন পাওয়া যায় না। সো হোয়াট they came up with the idea যে সকালবেলা আমরা বেরোবো, যেখানে বনের বাজারে শিম্পাঞ্জিদের বাসা যেখানে থাকে, সেখানে যেখানে শিম্পাঞ্জিদের পটি পড়ে আছে, we will collect that. From that we will isolate chimpanzee DNA and viral RNA. All right, so that is their job. It's a very difficult job that they have to isolate from fecal samples chimp DNA and viral RNA. They have to isolate. All right, তাহলে এটা একটা বিশাল বড় রিসার্চ, and they kept on doing it for several years. যখন রেজাল্টটা বেরোলো, it was stunning. দুটো জিনিস দেখা গেল। এক হচ্ছে এই ভাইরাসটা exists in wild chimpanzees. Yes, it does exist. তাদের কোন কোন শিম্পাঞ্জিদের আছে? বনোদের নেই, এই নাইজেরিয়া ক্যামেরুনিয়ান শিম্পাঞ্জিদের নেই, ওয়েস্টার্ন শিম্পাঞ্জিদের নেই। সেন্ট্রাল এবং ইস্টার্ন এই দুটো শিম্পাঞ্জি সাবস্পিসিস, দে ক্যারি দিস ভাইরাস, এবং দে ক্যারি, এদের দেখা যাচ্ছে শিম্পাঞ্জিরা যেরকম কমিউনিটিতে থাকে, some communities have 50% chimpanzees are infected. It doesn't seem খুব একটা অসুখ করায়, but they are carrying the virus. আরো ডিটেইলস বার করলেন, দেখো you can see this kind of paper থেকে, রিসার্চ আর্টিকেল থেকে নেওয়া, যেসব জায়গায় ভাইরাসটা পাওয়া গেছে সেখানে it is marked in, you can see these are the areas যেখানে ম্যাক্সিমাম ভাইরাসটা পাওয়া গেছে in chimpanzee population. অলরাইট, আরো করে দেখান, তাহলে কি দেখা গেল? Chimpanzee viruses found in wild chimpanzees, two subspecies of wild chimpanzees, they match the HIV-1 virus of chimpanzees. অনেক বেশি infected হয় than humans, but it does not seem to cause any major disease in them. All right, to a certain extent. সেখান থেকে কনক্লুশন কি হলো? Pan troglodytes, মানে the common chimpanzee is the natural host and long-term preserver of the simian immunodeficiency virus, which is the ancestral strain of HIV-1. তার মানে সেন্ট্রাল আফ্রিকার শিম্পাঞ্জিস থেকে the virus has jumped to human. How? Most likely ওই bushmeat trade. কারণ শিম্পাঞ্জিও প্রচুর ধরা হয়, মারা হয়, কাটা হয়, খাওয়া হয়. That is how it has most likely jumped into human. Last question. How did chimpanzees get the virus? How did chimpanzees get the virus? Because if you recall, most of our talk has been that এই ধরনের virus you get in African monkeys, right? How did chimpanzees get the virus? তখন দেখা গেল যে শিম্পাঞ্জিদের যে ভাইরাসটা আছে তার সঙ্গে দুটো আফ্রিকান মাঙ্কির ভাইরাস খুব ম্যাচ করছে। ওয়ান হচ্ছে রেড ক্যাপ ম্যাঙ্গাবে, আর একটা হচ্ছে এই স্পট নোজ মাঙ্কি। ওকে, সো এই দুটো মাঙ্কির যে ভাইরাসটা আছে, the chimpanzee virus very strongly resembles the virus of these two monkeys. In fact, শিম্পাঞ্জিদের যে ভাইরাসটা পাওয়া যায়, it is a hybrid of these two monkey viruses. All right, এবং রেঞ্জটা লক্ষ্য করো, আবার সেই ওয়েস্টার্ন আফ্রিকা, ওয়েস্টার্ন সেন্ট্রাল আফ্রিকা, ঠিক আছে? How did chimpanzees get these viruses? তার কারণ হচ্ছে শিম্পাঞ্জিরা eat these monkeys from time to time. Chimpanzees are mainly frugivorous, but from time to time they eat these monkey, hunt and eat these monkeys. এবং it has been tabulated যে শিম্পাঞ্জিরা যত ধরনের স্পিসিস শিকার করে খায়, ম্যাক্সিমাম হচ্ছে এই দুটো স্পিসিস, ওই ম্যাঙ্গাবে আর ওই স্পট নোজ মাঙ্কি। তাহলে কি দাঁড়াচ্ছে? শিম্পাঞ্জিরা got these viruses from hunting these monkeys, and humans got these viruses from hunting chimpanzees in the bushmeat trade. So the cross species transmission, evolution, cross species transmission, evolution, ভাইরাসটা কিরকম নতুন স্প্রেডিং ফর্ম হোক, বুঝা গেল? ও ভাইরাসেস দেখো সেম প্যাটার্নটা ম্যাচ করে যাচ্ছে। Where did it happen? Maximum chance যেখানে maximum infection of chimpanzees, that is in southern Cameroon. এই যেখানে লাল রঙের this MB, LB areas. So chimpanzees in southeastern and south central Cameroon are the sources of HIV-1 groups M and N respectively. All right. So this is where it all ends, and this is how the final phylogenetic tree looks like. HIV-2 সঙ্গে সুটি ম্যাঙ্গাবে ভাইরাসের মিল আছে, শিম্পাঞ্জির সঙ্গে ভাইরাসের সঙ্গে HIV-1 ভাইরাসগুলোর। All right. Aniruddha, do I have 2 minutes? All right. Thing is, শিম্পাঞ্জি থেকে এসছে দেখাইতে যাচ্ছে, সুটি ম্যাঙ্গাবে মাঙ্কি এসছে দেখা যাচ্ছে। If they can come from those, why not from others? All right. So what now scientists started doing? Abar oi potty analysis. All right. So what is that? Anyway, they were going to the forest and collecting pieces of chimpanzee. এবার সব সময় তো চেনা যায় না কোনটা কার পটি, অনেক সময় ভুল করে গরিলার শিম্পাঞ্জি পটিও নিয়ে নিচ্ছে। So they did analysis of that also, and they found that some gorillas were also having a virus, SIV-1, and this was closely matching HIV-1. এই ভাইরাসটা closely matching HIV group 4. তার মানে তিনটে HIV গ্রুপস আছে, MNP যেটা শিম্পাঞ্জি ভাইরাস থেকে এসেছে, আর একটা আছে O যেটা গরিলা ভাইরাস থেকে এসেছে। Again, gorillas most probably some kind of contamination with humans to rest. একটাই প্রশ্ন, গরিলারা এটা পেল, কারণ gorillas are strictly herbivores. All right, so how did they get that? Actually, nobody knows the answer. It must have happened in the forests of Cameroon or Guinea or Gabon. Most likely, the chimpanzees and gorillas stay in the same forests in many places. এখানে হয়তো some kind of fighting may have happened এবং then bites and scratches and cuts and virus may have spread. This is the most likely answer. How gorillas got. All right. So I think that is the story that I wanted to tell you. Class lesson. Yeah. That is the story I wanted to tell you. That how a virus which causes a pandemic, has jumped again and again from primate species to humans. But এমনিতে এতটা হতো না. It is mainly due to human activities. এই সাংঘাতিক বুশমিট ট্রেড না হলে এই জিনিসটা হওয়ার চান্স খুবই কম, right? So human activities কিন্তু can result in zoonosis. এবং লাস্টে I want to tell you one thing. আমাদের এই যে করোনা ভাইরাস প্যান্ডেমিক এবং আগের যেসব করোনা ভাইরাস যেসব প্যান্ডেমিক হয়েছে 2003 হয়েছে 2012 হয়েছে, all of those scientists believe একই রকম ভাবে from some animal in the wild it has spread to humans. এবং think about, ঠিক যেমন here we are talking of cities like Kinshasa, আমাদের প্যান্ডেমিকটা কোথায় প্রথম শুরু হয়েছিল? Wuhan, China. And Wuhan is a city which is larger than Calcutta. How millions of people live there. A virus which was going around maybe in rural China, যেই Wuhan-এ ঢুকলো সেটা তো আরো respiratory virus. It started spreading even faster. All right. So human activities can result in new zoonotic. All right. That's all I have to say. Thank you. So my major references, these are two very lovely books that you can read. And it is actually written for the general student. They are very nicely written. These are the other references that I got from. All right. All right. Thank you, Maniganda, for wonderful, very informative deliberation. Questions? Last questions to the speaker. সবকিছু একদম বুঝে গেছে 100% নিশ্চয়ই না?
How likely is it that besides Wuhan, some other center would have been the center for I mean, is there anything special about any sort of human activity in Wuhan that was the epicenter or?
See, thing is that there are two things here. One is the scientific part, the other is the political part. And the entire scientific part has got completely messed up because of US versus China geopolitics. Correct. So that is the other problem. The problem with the thing is that Wuhan is a city যেখানে lots of people are there obviously. Plus, more recently যেগুলো দেখা যাচ্ছে that ওখানে যেসব animal markets আছে যেগুলো মোটামুটি আমাদের বাজারের মতো. So those are places where animals like these are stored পাশাপাশি. এবং scientists have actually gone to those markets and isolated SARS-CoV-2 RNA from those sources. এবং সেখানে একটা kind of widely I think believed in government circles that the virus was probably present in southern rural China for quite some time. কারণ এই ধরনের viruses SARS related coronaviruses, they are found all across China south, পুরো Southeast Asia জুড়ে, Laos, সব জায়গায় পাওয়া যাচ্ছে. Okay, Thailand, everywhere it's being discovered, all these kind of viruses. Thing is that these viruses ওই অ্যানিমালসদের দিয়ে they could have traveled to Wuhan কারণ এই বিশাল অ্যানিমাল মিট ট্রেডটা চলছে এবং সেখানে যখন once it gets a dense area যেখানে সব ধরনের ভাইরাস একসঙ্গে আছে, so it is quite likely এটা একটা possibility. The other possibility is that চায়নাতে আমি এটা আগেও বলেছিলাম সেটা হচ্ছে যে চায়নাতে there is now this big business of মানে ওই যে পশমের যে কোট গুলো হয় ইটস এ ভেরি বিগ মানে হিউজ ইন্টারন্যাশনাল বিজনেস এবং the fur actually comes from the animal যাকে বলে raccoon dogs মানে ছোট একটা শেয়ালের মতো দেখতে গায়ে খুব লোম আছে হ্যাঁ raccoon dogs গুলো ওর থেকে আসে না চায়নাতে এই raccoon dogs এর ফার্মস বিশাল বিশাল রয়েছে এই ফার্মস গুলো কোথায় শহরের মানে শহর আর গ্রামের জাস্ট ইন্টারফেস এবং in these farms thousands of animals stay together একদম closely packed এই many of these farms are actually near forest areas এবার যেই forest areas সেখানে চান্স আছে সেখানে অনেক বাদুড় আছে বাদুড় that's the source of many of actually sulfide original source ছিল বাদুড় so these বাদুড়রা হয়তো ছিল কোনো একটা either some kind of saliva অথবা hospital কিছু একটা করেছে সেগুলো এই অ্যানিমালসরা খেয়েছে এইবারে the other thing maybe সব স্পেকুলেটিভ কারণ 2003 যে SARS ফার্স্ট প্যান্ডেমিক সেটার সময় বেরিয়েছে যে raccoon dogs are the one of the major sources of SARS virus. They got it from bats and it has spread to humans from raccoon dogs. কেন one of the things হচ্ছে raccoon dogs কে মারা হয় for animal meat নাম্বার 1 নাম্বার 2 হচ্ছে যখন এই ফারটা গাছ থেকে তুলে নেওয়া হয় তখন জন্তুগুলো চেঁচায় মানে একটা ধস্তা ধস্তি সবকিছু একটা হচ্ছে তাহলে হাই চান্স একটা রেসপিরেটরি ভাইরাস স্প্রেড করে যাচ্ছে so these are believed to be good possibilities of how the virus could have started to spread মুশকিল হয়ে গেছে চায়নাও বলবে না ইউএসও বলবে না পুরো একদম জিনিসটা পুরো গেটে গেছে তার মধ্যে the whole idea of epidemiology হচ্ছে যে viruses usually follow these kind of zoonotic path so যেটা আগে হয়েছে সেটা আবার হবার পাইছে এবার চায়না বলবে না কারণ তাদের ফার মিট ফার ট্রেড বন্ধ করতে হবে যারা ফার কেনে তারাও বলবে না
থ্যাংক ইউ স্যার Anyone else has any question?
It's not actually a question. I am just curious to know that since it's proved that most of the viruses are coming from animals, so can it be a preventive measure that whenever we found that some city is a center of epidemic, so can it be a preventive measure that we just stop eating meats or become vegetarian for some period to stop spreading the viruses for some period?
সেটা তো মানে ফর এক্সাম্পল দেখবেন যখনই from time to time যখন flu রেগুলো আসে people say now they have a lot of caution in eating meat and things like that possible you can মানে say on বর্ষাকালে you boil the egg properly and eat half boiled খেও না see obviously those can be done problem হচ্ছে as far as I understand এগুলো হচ্ছে কিন্তু at the very personal level you can't really stop unless it is a government policy that we are going to stop this kind of illegal animal trade না হলে আমি আপনি খেলাম না সেটা there's an economic driving it right
That's why I'm asking that there is no actually guidelines in that time. Only from newspaper we are actually we get to know that the virus is spreading.
but in that case in case of the coronavirus respiratory virus so that thing would not really hold in fact মানে initially নিশ্চয়ই সবার মনে আছে যে আমরা সারাক্ষণ হাত ধুচ্ছিলাম হ্যাঁ so পরে দেখা গেল that it is not spreading through surfaces also in fact জিনিসটা became very strange যারা মাস্ক পরছে না তারাও স্যানিটাইজারে হাত ধুচ্ছে মানে why do you do that মানে একটা রেসপিরেটরি ভাইরাস আটকাতে হবে but that was the initial guideline পরে আর WHO guideline চেঞ্জ করেছে so those kind of things but yes disease specific guidelines question হচ্ছে যে how fast you can bring them আজকে অসুখটা সরকার আরম্ভ করছে আপনি এক মাস পর মিটিং হলো তখন বললেন যে গাইডলাইন
Just feel.
Straight.
Very difficult. You need a very fast strategy.
সেটা only the political leadership can do if they are listening to people who know the facts. Who know the facts, know the that's all. But it applies many times. Oh, no, music. Initially started now. It actually start howar por. Actually, there were certain bans on certain kinds of foods and things. It was done. Trouble is, over time, সেই ব্যাংকগুলো আর কাজ করে না, especially when it's a lot of millions of dollars, you will find it there. But certain things were done এবং from 2006 আমার কাছে একটা পেপার ছিল আমি দেখা from 2006 কনফারেন্সেতে সায়েন্টিস্টরা বলছেন যে if SARS virus can come like this, can again come. But then who listens to scientists and teachers? Isn't it? Nothing like that. Okay. So it's been said from 2006. They acted about, but people have not listened. Once you have listened. That's it. All right. Any other questions? Anybody any questions you can ask without any hesitation. একদম কোনো অসুবিধা নেই।
লজ্জা পাওয়ার ভয় পাওয়ার কিছু
হ্যাঁ লজ্জা পাবার কিছু নেই। ও লজ্জা আমরাও পেতাম সেটা কেটে গেছে সেটা আমি জিজ্ঞেস করতে পারবো।
There are no further questions.
স্যার যদি আবার করোনা ভাইরাস আসে তাহলে এখন থেকে কোনো প্রিভেন্টিভ মেজার্স নেওয়া কি পসিবল? আর কিছু দরকার আছে
না কেন সেটি হচ্ছে
কোনো মেজার সেভাবে হসপিটাল যেটা
দেখো পরে রেসপিরেটরি প্যাথোজেন measures acted. Say avoid as much as possible, avoid indoor crowding, and if you have to be there, wear masks as much as possible. Respiratory pathogen to me, I mean, you have to do something which is practical. So respiratory pathogen maximum in the bite is have to not right? Where can you wear a mask? Say to me bank is there. ব্যাংকে সারাদিন অনেক লোক আসছে এবং তারা চলে গেলেও ব্যাংকটায় এসি বলে সব জানলা দরজা বন্ধ ভাইরাস বিকজ ওয়ান্স ইনসাইড স্টিল সার্কুলেটিং ইনসাইড সো ইউ ক্যান এট দ্যাট টাইম যে তুমি 15 মিনিট থাকলে ইউ ক্যান ওয়্যার ইট বিসাইডস দ্যাট হোয়াট এলস ক্যান ইউ ডু ইউ ক্যান ওয়েট ফর
it's a session for the marks for it's very suffocating at the same time. তারপরে
Yes.
humidity ও হয়ে গেল তাহলে তো সব humidity তে যেভাবে হয়ে গেল তো it's very
these are problems that after moving disease patient beyond the point we have to face. There is always the risk, especially for a disease like COVID, that does a lot of damage to the cardiovascular system. We are in bigger thing. There is no doubt. Question is, কিছুটা কমানো যায় কিছু না করার থেকে একটুখানি করাবো obviously তোমার যদি শ্বাসকষ্ট হয় নিশ্চয়ই করবে না তাহলে তো অপারেট হয়ে যাবে isn't it? But if it is not, for some time if it is very good, danger is there. The virus is very much there.
Absolutely. What? Yes. There are no further questions. Once again, I thank Dr. Yogananda for that. It was a very great thing. So we will keep hearing other talks from me in future also. Next speaker is going to give his talk online. Renewable energies. I have the privilege of working for a long time. It was not a passion to get you actually. Later he went on to biology and is a practicing biologist right now. So before we beginning stop, like you went to on and introduce the speaker. Am I audible? Okay.
Thank you, Anurindra. Uh, Good afternoon, to our respected principals, coordinator of IQAC, CW, to my learned colleagues, distinguished participants, and students. It is an honor for me to introduce Dr. Inul Banerjee. Dr. Banerjee completed his post-graduation from Indian Institute of Technology, IIT Kanpur, in Biological Science and Bioengineering. Then he did his doctoral studies in Institute of Biochemistry, Swiss Federal Institute of Technology, Switzerland. Subsequently, he served as postdoctoral fellow in Institute of Biochemistry, Swiss Federal Institute, and the Friedrich Nietzsche Institute in Basel, and Novartis Institute of Biomedical Research, both in Switzerland. Presently, he is an assistant professor in the Department of Biological Sciences, Kaiser Mohali. He is a biologist, has published his works in various prestigious journals, including the journal Science. On the behalf of the faculty of Department of Zoology, SMCW, I welcome Dr. Henry Banerjee. Dr. Banerjee, the platform is yours.
Thank you for this very kind introduction. Am I audible to others? Am I audible? So, thank you again. Like, start by thanking the organizers, actually, all the with my friend for a long time, and Dr. Kapri Dutta for sending this invite. Which will share speakers. Introduction to my lab. Our lab is one of the cellular biology lab, or we call CP lab. It's now been recognized as scientific transmission by CMR and recognized in Mohali. And since the short form of our lab is CP lab, we call everyone as CB. So any of you want to hear CB vision, easier, the most welcome to apply flights in our consumption channel. So talk about later in the talk. So to begin with, let me introduce influenza virus. So we work on three different viruses. That is influenza virus, or specifically influenza A virus, or IAV, SARS-CoV-2, and dengue. Three medically important viruses. But in today's talk, I majorly focus on the biology of influenza A virus, how the virus evolves, how the tropism of the virus changes, how the virus spreads from animals over humans, and what are the good needs to tackle the viral infection. So to begin with influenza virus, influenza A virus, it is commonly recognized flu. It's an infectious disease which is caused by an RNA virus, which is single-strand virus. And this virus belongs to the family of Coniferinae, and it majorly affects birds and mammals. If you go for the classification, there are five genera of this Optomyxoid family, and there are influenza A, B, C, and very recently classified as E as well, coronavirus is a virus. And among them, influenza A virus is the most usually form. But influenza B virus, which is called IBV, can also infect humans, but they do not cause much trouble in humans. To look at the structure of the virus, it's very simple, it's very tiny, about 100 nanometer in diameter, and it is an envelope. And this envelope is provided by the host in which the virus provides. And just below its envelope, there's an capsid which protects the viral genome. I'm going to pictures of the structure. And as you can see here, there are other proteins of which are going to the viral surface, which are called the protein, immunoglobulins, and there's a channel that's called the protein channel. And of course, there are viral genome inside. So if you look at the virus structure, this is how the virus looks like. It's not real. So I try to make the structure from primary images into components of this. We have now constituted in 3D. As you can see here, that there's a multiplication, which is in the surface. And this is the most pattern protein on the virus surface. The virus uses it for two purposes. One is for its attachment to the cell receptor. Of course, if the virus wants to infect cell, it needs to find its target receptor on the host cell. And this is like the lock key. So the key is provided by the virus, and the lock is present on our body, our cell, at the cell surface. This is called the receptor key. So with this key, the virus first tries to unlock the door, and this is primarily done by this protein. But when it enters the cell, the virus has to breach the membrane barrier. That is, the virus is in the cytoplasm, it's minimally involved. The virus is still interacting with these problems. The virus needs to come out. How to come out? The virus uses the same key that it used to enter the cell to breach the membrane barrier. So this is all viral goes to immunoglobulins. Serves two purposes. One is attachment to cell receptor as well as validation when the virus is already evidence. The next is channel, like the neutral ion channel, which is responsible for the passage of photons from the environment, that's from cell, to the viral form. Why this is necessary? When the virus is entering the cell, virus capsulates the resistant. And we know that for the resistant to water, there needs there is a need for some ion changes which would destabilize the capsule. And this is brought about by using photons which are entering the viral form. And this is mediated by this channel. And then there's another protein which is in the cell surface called immunoglobulins. What is the function of immunoglobulins? When the viruses are finally leaving the cell, they're still attached to the cell. So immunoglobulins is an enzyme which gives off the interactions between the virus and the surface. So this is involved in virus release. And inside the virus, you can see for influenza, there are 8 segments in the viral gene. Because the SARS-CoV-2 is a very long RNA break of 32 progresses. But in influenza, it is more complicated. The entire genome has been divided into 8 segments. And if you look at this VRNA, this is called viral proteins, and it is packed with the viral protein called and at the end, you can see the viral polymerase. So the virus comes with a package from cell or protein tissue, and this is called polymerase complex. Now coming to host specificity. So as Dr. Mitchell said, that for most of the viruses, the reservoir is wild animals. For influenza, it is a wild birds. Short birds or eat the waterfowls. So here, the viruses are circulating in these animals, in these birds, but they are not causing sickness. So the hosts are very happy with the virus, and the hosts are in there. That's wild animals. Sometimes, when the wild aquatic birds come into contact with the healthy birds, say ducks or chickens, the virus can be transmitted from the wild aquatic birds to humans. And when the multiverse makes some domesticated animals like pigs, there will be a spillover from multiverse pigs. Occasionally, pigs can also be infected with the virus from wild pig birds. But remember, in general, I'm saying in general, not specifically, that the avian influenza viruses cannot infect humans. Why? I'm going to later. But from pigs, humans can be infected. So I can also come to the birds. So what is the difference? Why the wild viruses which have infected or circulated the wild birds cannot directly humans? Here lies as probably the details I just like mentioned, the peak, as you can see here, serves as an excellent vector for mixing up different kinds of viruses. So pigs can get infected with the influenza virus. The influenza virus is 7.4 fevers, and it can also be infected by humans. So all sorts of influenza viruses can be infected. Therefore, pig is called excellent mixing the so-called viruses. Now coming to the specificity of different viruses in their cells. Influenza virus, as I told you, at first it needs to find its the receptor on the surface. And the key receptor for influenza is silent. It's a sugar. It's a powder of sugar on the like fluids of like levels. And there's a specificity. So the hemoglobin protein that is present on the virus surface of 18 only can bind to the terminal salic acid receptor alpha 2. Because of fever, it is alpha 2 system. So there are some proteins, some amino acids which confer the specificity of AG in the salic acid. So the avian can infect avian viruses can attach to the receptor which has the alpha linked salic acid, and the human influenza viruses can only attach to the receptors covering the alpha 2 sites. In case of pigs, we find that both the salic receptors are present, and therefore the pigs can be infected by both the viruses, and therefore serves as a resiliency of the virus. Now, in case of avian viruses, we sometimes term this words part 2 has profile. So how does avian influenza virus can infect humans? They can only bind to salic acids alpha-3 which is mostly absent in the respiratory tract of humans. So how can it infect? The viruses are continuously mutated while they're circulating, not while they're living also. And because of this mutation, some of the mutations can have function. And because of this gain of function, they can change their function. So the same mercury molecule that usually binds to alpha-salic acids now can bind to both alpha-2 and alpha-6. And once it becomes complicated in recognizing alpha-2 linked salic acids, then you can also experience. So the changes from alpha-2 to alpha-6, alpha-2 again, really alpha-2 is present in first sequence. As it mutates and it explains the function, it can now recognize alpha-6 and it can infect humans. And this is how the transmission happens from proteins to humans. And of course, when the virus enters the human body for influenza virus, initially it's restricted to the upper respiratory tract, but sometimes the virus spreads down to the lower tract and causes severe infection. So, now there's another level of complexity. So far, I have talked about one conditions which make the viruses capable of changing its sources. But I told you that the viral genome is viral sequence. So what happens when two different viruses enter commonwealth, that is big. It's a very interesting situation that we see here one virus is connected to the flu genome and another is the genome. But one human origin and the human origin. And when they infect a pig, which is mixing vessel, while the viruses are crisp when the protein virus producing, the viruses cannot discriminate its parents, right? So there happens even a piece of so the segments from one virus it hits a good segments from the other virus, and the protein viruses can have the segments from season different. So reassault and reassault drastically changes the makeup of the virus. And reassortment is cause for major pandemics for interest. So there would be quite preventions and there would be just there genetic makeup is solved, there's a chip and finally this is over here. As you can see here, there has been there is several pandemics over here. So first because of pandemic which caused vast across the globe, even more than people all over the world. Was the Spanish flu broke out in 1988. And then there's a nation group, which is this engine, and this is caused by reassortment. So the first flu, the pandemic flu, that was the Spanish flu, that one 1918 flu, that was caused by single mutation when the viruses acquired the tendency to infect humans. And then this increased the subsequent pandemics. All the pandemics were caused by reassortment. So two different viruses, we want to take in origin, human origin, they are getting mixed up and they are spilling over into the population, and humans were not prepared to tackle the viruses, and there was a lot of death caused one, and the very recent one was 2009 pandemic, which was caused by one Lincoln variant. It was also reassorted virus. Whenever we talk about viruses, we only think of the viral genetics. But imagine a situation that the virus is infecting, say, 100 individuals. I'm talking about humans now. And what we see that most of the humans are asymptomatic. They do not show something. Maybe 8 to 10% of humans show mild symptom, and only very few percent show very severe symptom. Despite being exposed to the virus, the same case, the symptoms vary. What is the cause? There could be two causes. One is an environmental cause, one is a physical cause. Here, I'm talking about the genetics of humans. So there could be many environmental factors by exposure to the virus. Someone was really getting exposed to one strain or one particular group of viruses, and he has enough antibodies to which provide the protection against new viruses. Pregnancy, obesity, age, pregnant. And genetic factors, we have humans have about 25 million out of the 26,000 genes. And we are all different because we have small mutations on the genes. And there are some genes which are involved in RNA. And the first important question is, has recently been revealed to play a very important role in the outcome of the disease. So at present, I might be infected with it because I don't know what's the virus, but I'm not really symptom. Why? Because either my genes are taking care of infections, or I have been already exposed to this virus so that I have enough antibody in my blood to neutralize the virus. This is better. So, of course, genetics plays a great role. So, when search for business, a profile like many people who search to know what associations screen or anything else like that. Association screen of strategies. And what they found that we have the small mutations, and some of the mutations are associated with the disease, and some are associated with the severe form of the disease. So these small mutations are called SNPs or single polymorphisms. Why we call it polymorphism? Because this polymorphisms are present in the mutation. They're extremely rare. For example, here there are some sleeps which are present in if you reach the case or take the or selling. You find in the normal population, you find that three cells. So in case of T, to be a G, or in case of A, if you have a C. But we are all fine. We are not showing any symptom. This is very because we have hypothesis. It's really that's what was. But when it comes to viral infection, the small mutations can cause problem. The small mutations can determine whether a person who remains uninfected or a person is infected but should no symptom or should mild symptom or a person with progressive severe conditions. And this is what we did when in the early days of COVID. So we screened for all the association studies, given candidate studies, and we looked for the polymorphisms. And we found that there are 31 genes which are responsible for causing resistance or causing or pushing. See, if I study one genes and found polymorphisms, so we put that we have found 22,000 patients and more than 4 lakh, almost 4 lakh full subjects. So we were identified and find team stems of the time and this feature for the email Canberra performance see that we identified from very. So as I told you that first plays a big role in determining the final outcome. Now, coming to one very critical question. So how does virus here I'm an example for example in the first setting. So when the virus encounters the cell first needs to find its receptor on the surface. So this is called virus binding. Here you can see a virus binding its receptor point in terms of its EGFR and some other receptors. So what I'm not going to do is once the virus is catching the surface, then the virus is to enter the surface. There's a problem. What is the problem? Is the cell has plasmid, which for the viruses, for many of the viruses, is very difficult to reach. This is a very cell fails the virus. So the virus cannot reach the area. So what it does, something. It's able to hijacks the cellular inside tissue. And the cell, for its survival, needs signals and nutrients from the environment, right? So all the nutrients, the hormones, so they get concentrated on the cell surface and the cell engulfs those nutrients. So this is a process of endocytosis, and the virus uses this process of endocytosis to develop this. So it has entered the ribosome, it is called an RNA. And then the ribosome matrix is something called an ribosome where it's acid. And this acid environment of the ribosome serves as a key for the virus to drastically change the information of the surface protein. That is agent. Remember, beginning I told you that agent protein serves two purposes. One is attachment, and the second is fusion. And here, in the acidic environment of the living ozone virus, Lychee undergoes plastic condition in its machine and then fuses with the invisible movement. And how we shoot the viral contains. This is what the virus wants to release its genome into the cell so that it can now replicate and make protein viruses. For most of the viruses, the genome stays in cytoplasmic, but for influenza, the genome exists in and upon entering this, it either undergoes transcription or mutation, and the virus makes its mRNA proteins or proteins re-enter the nucleus, and new mRNAs of the viral genetic materials are assembled. And this newly assembled viral genetic material is exported out of the nucleus and get transferred to a cell plasmid. And some of the viral proteins are synthesized in pathology and they're transferred across the transpathology and they're deposited on the cell. And this is called the viral disease cell. And from here, the viruses follow infect the surrounding cells around it. So the virus isn't inert material. It has some genetic material, some proteins, and an envelope, but it is unable to perform these functions if there is substantial amount of assistance from the cell virus. And these are the host factors, human host factors, which are actually associated the virus to accomplish its job at different stages. And virus can see from attachment to the of the viral genome is an attractive target for viral vector. Because if you stop the viral entry, you can stop all the subsequent processes. That is, you can transfer of the viral proteins, plasma membrane, and viral part. You can stop the process, you can target the immune process. The other advantage is that many viruses use the same pathways to enter the cell. For example, influenza or SARS-CoV-2 use a vacuum-mediated pathway into the cell, and so the same virus. So you can target simple pathways. It can be possible to have a broad spectrum attack or to broadly attenuate the virus state. Now, how to monitor the virus? Very quickly, I'll go through it. But as I told you, the virus can enter by the process of cytosines, and there are multiple endocytosis pathways for the pathways indicated. One is capillary pathway, another is endocytosis, this one is cell-free. The virus, influenza virus, is this pathway right? And you can see here, because the microscopy frequency is a control microscopy. You can see the green dots inside the cell in the gray area. So these green dots indicate the viruses, the virus is already creating a virus. And you can also see some red dots around the cell surface or around the cell feature. These green dots of viruses feature outside the cell. So non-humanized viruses versus humanized virus. You can have this assay to track the virus in vivo. And now if we look at the PDF, you can see from this is the resolution. Imaging actually the viruses are big. It's viruses in the person type. So you can see here within the endosome the viruses are injured. You can see one virus is centers and you can see one there inside the as I told you that if they enter the endosome, it is very difficult for the virus to exit the endosome. So the virus strategy, and that is what? That is the conformational change of the AJ, which makes it fusion complexity. In the surface AJ, this is the surface AJ in equal conditions, but as I told you, as the endosome matures, the pH drops and it's a drastic confusion change for AJ. As you can see here, there are some great lines to competition which in vivo pH is very quickly, but when the virus is exposed, the one pH fusion peptides residence. And this fusion peptide then inserts into the fluid membrane of the endosome, which lies as fusion. And here you can see the green box against yes, we can antibody like a targets the form of you can detect the virus is acidified. If you block the acidity of the endosome by different frequency methods, and you get the you do not see acidified virus. So you can stop the virus infection at any stage, starting from binding, endocytosis, acidification, then comes unfolding here. After fusion, the viral capsid resistance, releasing its genome, and you can see first the syndrome of the viral capsid, and after that, the virus enters the viral nucleus. You can see here in this picture, in DNA-circular cells that is encode the viral proteins, the viral genome the nucleus, but the drug which inhibits the transport of the viral genome to the nucleus. You can see the viruses are the surrounding region of the nucleus are unable to enter the nucleus. Okay, so here I'd like to how the viruses infect other course time. So at 3 hours, then hardly take the viral infected cells. You see some amount of nucleotide entry in the nucleus. As it is progressing, 6 hours, you can see the emphasis of getting brighter. That means the virus is producing more and more of this nucleotide. At 8 hours, you can see the lake protein that is included is produced, and you see very strong signal. The nuclear area is bulging out, so the virus is producing this protein in the Golgi brothers, and you see the inventory is very bright. The nucleus, the virus is using a lot of viruses, not protein viruses and antibodies. And at the late hour, that is at 12 hours, you see that the signal intensity of infinity is reducing inside the nuclear area, but you can see it in the cytoplasmic area. So the virus is now exiting the nucleus and entering the cytoplasm for what? For packaging of the ingredients and for virus virus. And you see 24 hours to see collection. You can see there's hardly any nuclear protein inside the nucleus, but it's usually present in the cytosol. Humidity is present all over the cytosol, and you can see how the viruses are coming off. It's like their lines. These are the predictions which the viruses make from the host cell to make protein. So far, I've given you the brief overview of the cell biology influenced virus. Now, problems. So far, all the antivirals which are present currently out and out are godly virus. I know that the virus is increasingly mutated, and the viruses like influenza, SARS-CoV-2, these are all other viruses which are extremely prone to mutation. So whenever we are making a drug and taking out the market, which takes about 10 to 12 years and involves billions of dollars, and after it is finally out of the market, we see the viruses are either live or they are mutated of the tissues. So there are three compounds which are active, that is specifically the zolanomid granules, coming which is heavily used in case of food. And there's some resistance important for solve. People oseltamivir or selamivir, there's another group of compounds, drugs which are used, this amivir like lamivir. But the viruses mutated so fast, like it was in 2030, they were almost resistant, or 100% resistant in just a So their clinical use has been discontinued since 2012. There's of course a new compound that is peroxidine, but it is also reported that that was received. And that's also resistance. So growing antiviral resistance against influenza is a major concern. So what would be the alternate strategy? So the alternate strategy that we are taking is post-influenza. I told you that it is not only the virus, but the host. And the interaction between the virus and the host. So can we not target some of the hosts which are spiked by the virus or virus? I told you that virus is an extremely complicated process, and virus interacts heavily with the host proteins. Here you can see the viral proteins interacting with host protein, but the list is not exhaustive. It's interacting far more proteins than we thought, and because that's of the host factors, at every step of its lifetime. So if you're talking of perspectives, there's advantages and disadvantages. The advantages are first factors are unlike is the host, and all the host proteins are derived from the host DNA, and so we are unlike the virus viral proteins us. And the host proteins are unlike in these final nucleons and long-term because virus they want to infect the host cell, they have to infect us. And if you silence the host vectors, the virus not infect the host at all. Another contributors. There are only a few first factors which are exploited by the virus. So, if we can target the key host factors which are exploited by multiple viruses, we can actually uh, get a broad spectrum antigen. So this is our strategy. Of course, if basically targeting a host, this is your essential functions, and everything on that side. So what is the challenge? Challenge is to find a specific compound with high immunity against virus, but with some with no or tolerance. So that is an idea. For this, I just very quickly attend the story of the environmental discovery. Like, how do you know that? And so what we did, we experienced in small molecules against influenza. This is a high fidelity microscopy system that we have. Something the photo microscope in which we can perform 384 experiments in a single shot using 384 paper lengths. So the first thing you see the cells, you see the one in the cells and the space, and we treat the cells with the compounds for about an hour. Then we inspect the influenza virus, then this is antibodies. First, the antibodies you can identify the cells from very effective. So here you can see that increasing what is coming from the viral infected cells, and we get the CDK. And we screened a set of like 89 compounds. I'm sure the set of the compounds are tested. And here you can see that there's one compound 27. So this is a map where it indicates 100% emission. And it's a really slow emission. So here, the surprising found one compound which negative viral protection contributes. I usually contributes because it is too it's very unlikely that you will find how you can viral protection. I was very skeptical, but that's first experiments. So when you look at it about 20 times, each time we hope that he found the machine, how much he found. So it was unplanned, it was unrealized. So we then looked into the compound. This is compound. It's a very simple compound. You can see there's a urea in the middle, and the urea is going through behind. And on the cylinders, you can see state figures. And this compound was electron-crazy compound, which does not contain the state figures. It's sort of like the LPS view, and we call this compound. It is. The only difference is presence and active substance. So the effect that we saw is the first one, right? That is having this figure. Now we press whether the parental compound also has an effect. So we tested the parental compound. It's present. It's present. The moment it has a C groups at 4 different corners of this molecule. So it's all or none. Then we got twisted, and we made several other analogs. This in terms of the constraints, it's in page 101. And here is a set of illusion. You can see that we find 5 compounds. It's from a quickly integrated question. And these are in microscope images, you can see the purity of the cells is hardly in the cell which is infected by this virus. And this is viral registration. I'm not going to all this stuff. So you'll see some data, but don't get nervous or simply report. You can see it's a Western law of the viral thing where you can see that the controls there's a deep magnetic band that means viral continuous press but in the cells where if you're with the compounds there's no band that keeps viruses were unable to replicate. And we look into viral the answer. Uh, we also calculated many things that are how maximum consistent terms for elasticity. So what is important is this that whether a compound is good enough to be regarded as a initial drug can be read. It's also index. And what we need is remember that needs for any compound having a selective index and more than 10 is considered as a good candidate for drug treatment. And you will see here the numbers for the compounds that got, like all of them show more than 20 compounds, but more than 30. Some, like the physical short percent, how. The study that this compounds are extremely good like this. But now we also did animal trials. We observed a very nice one in the recovery, survival. I'll go past here. So once we can address that one of the problems of the systemic violence is the induction of resistance. Right? So the drugs which would suppress the can one side, but if the virus is mutant, so that this more significantly change the virus its property, then this compound will be negative. And so, in order to check whether the compounds that we discovered can overcome this challenge, so we make a viral condition in the lab. So what we did, we considered the CC virus, and we took the commercially available surfactants resulted in less people. As you can see here in the very beginning, well, we need one active virus. But after we test the virus for 10 facilities, that is really like evolve the viruses. Then the virus was substantially weaker. And probably surprisingly found is that the viruses mutated in such a way that they became resistant against the clinically approved drug, which is used all over the world, that is something. But with absolutely zero resistance against bounce back, this one is here, this red. Then we went ahead and we tested another important virus in SARS-CoV-2. Here you can see there is a third panel coming here, and we tested the same compounds. And very surprisingly, found the compounds which were effective against influenza efficiently block SARS-CoV-2 as well. You see the same panel of mutations matched. And this indicated that this compounds are helping and these are very good results. So then again, this is that will be done with this is all very common. That's very good. And we tested with against different strains, the delta, omicrons, etc. So this were very much effective. We did animal trial, we did nucleus survival. And here for SARS-CoV-2, we use monotherapy. It's drug against SARS-CoV-2 before. And here we see the lung sections of the mice infected with the virus. And you see in the cells there's a lot of courage, increasing of the immune cells. But monotherapy treatment partially restored or partially recovered the infection. But if you compare symptom concept in one of the three, we saw remarkable clearance of the lungs from viruses. And we get that long story is higher. So we are very excited. Now we press the PC in the viral cycle. This should be destroyed. So we use the same acids, that is a virus binding type for the spike form, and found that these compounds do not interfere with the binding of the viruses. But what is very striking is when you look at the internalized virus particles, it's a remarkable increase. The number of virus particles which enters the cell with resistance powers. That is telling this compounds the targeting virus in all of these compounds. And when all subsequent states, virus that is the virus classification, virus controlling, virus solution, and viral state, all the viral entry states are blocked. So this is to summarize the message spark. And we change the entry of cells to be group, like influenza virus is to be group in people's block. And we also found interestingly that just a pre-treatment of the compounds for 2 hours, and then you remove the compound, this was sufficient to remove the virus. So this compounds can act as prophylactic as well. Preventive and prophylactic both. Now, if you look at these videos, you will see in the tumors of different vesicle cells, the rates dots of the viruses in dots like that, in the dots. The viruses are able to infect cells and they're very nice to recognize and data chromosomes, but in cubic cells, the viruses are arrested, cells surface, the movement is arrested, and then they are just on the cell surface, but they're unable to invade the cell. So here, vampires in chromosome marker, you can see the viruses are nice arrested at the cell surface in the presence of the compounds. And then we tested cells and we found cells it's not where you cell with presence of compounds. So we address how we use properly. Here I'll go very quickly the uses of funding results and the use of this right results of this virus is recently on the entry part and this virus is integrated with the classification of the radiosome. I'll just very quickly go through it and what we found by seeing is that these compounds are capable of bringing chloride ions and chloride homeostasis plays a critical role in the split dynamic, that is the homeostasis. And what we found in the chloride sensor, we encoded chloride sensor itself, that this compound is bringing in a lot of chloride ions inside the cell, thereby destabilizing the chloride equilibrium, which in turn is interfering with the vesicular dynamic. This is a pathway which the virus utilizes for its saving. Also, we found that because of the defects in the CP pathway, the surface receptors for the viruses, for influenza is CPFR or the subsequent SPF, their recycling is also purged. Here you can see in different cells that the silent is CPFR with the viral receptors, CPFR is present all over the cells. And it can be great if you use the transferrin receptor. But in case of cells with the compounds, you see there's a big accumulation of NGFR. Indicating that NGFR is entering the cell, or they're synthesized in the cell, but they're unable to get recycled back. So the number of receptors that are present in the plasma is low. And in case of SARS-CoV-2, that's the case in the receptor for this virus. And we see a dramatic increase in the number of receptors present in the cells. So to summarize the part, what we found that in the control cells, the virus can enter perfectly fine cells of the stress receptor, then it enters zoonoses, mechanism, uses the mechanism, then it releases capsid into the cytosol, and this is going this and immediately in itself, so the virus binds to the cell surface and is unable to enter. What it does, it breaks the chloride and this chloride accumulation destabilizes the intestinal fluid, which also interferes with the recycling of the receptors. So you see that there is less receptor, viral receptors on the cell surface. And there's accumulation of static pool of liposomes around the area. This is how the So this was we published a story, and so we also filed a press patent, and it was their new newspapers and also this televisions that are and this trade newspapers, it's legal, that's there are others. Like we are really excited about it, and we got quite a generous funding from Direct. It's a government grant agency which supports just such studies we have translation and initial. Comparative group testing as the policy. So because I come to the end of my talk, this is near Houston, which is Aizer Mohali, it's a community campus, we are the most welcome to visit campus, and these are some places of Aizer Mohali. And for those who are interested to know about more about the programs that we offer, we offer the regular business program, 5-year program, which is to create refugees and joint interns, IED. We also have the vertical exclusive program. This would be of interest to those who are pursuing bachelor's at present. So we have students of previously need to qualify one of the exams that I am paid. We are very selective about the students those who are really good for like the potential researcher. There's no fixed number of good for the students that they share. Then applies here, August and January. We also have a very good music program. So it's offer Greece or paper on US, paper on the club, and these students qualify one of the national exams. So we need to create some inspired. We also have a postdoctoral program where comes for from healing. And with knowledge, the SWOT, which I showed it, was done in collaboration with scientists from answer. Sorry, I Spengo, I Whipper, since I think here, there's a Latin, who's a person with this collaboration, my Whipper, is a organic size of 1 kilos, or 100 to have this if there is experiments. Is something done. And this one was literally spearheaded by student. Then it's now this Dr. Lino Mark is simply so we work on different aspects. I've just shown one aspect, one translation aspect that is going on in our lab, but we're not going to do because as I told you that we are from civilians, we also have a lot of fun together, but runners, these are the lab members, fast press members, interview with, and are interviewing with all of sir. They're most of them, or all of them are they're doing excellent job at extremely very students. I don't think no one should come to the lab or you know like for all well, sometimes I found few lines that details they're all prepared. I mean, there's no not timings, so they're real, but I have observed I have trusted. That's the kind of all of them are big pieces of my site visits. And now, last but not least, my the contingencies we have very numerous fundings that I said those startups, plants, then our assistant, journalists, Mr. Son, by the price of Mohammed, by web, with this, I thank you for your attention. I'll be very happy to take questions starting from the students. Thank you very much. Thank you very much. Let's send it all to the radio. Yeah, no questions. So, If you have any general question, not related to this topic, that is fine. In the search function that you also let's see now. You have responded a lot that you need the virus. You also tested that was only involvement virus testing. Usually the most
Dr. Banerjee, can you hear this? Yeah. And what I want to ask is more of the general question is how do you look at the news of the influenza reported from the United States laws nowadays? In the crowds and the flu news which is coming from the United States a lot recently. And any plans for extending your studies to those kind of strains?
Absolutely, would love to. So we have tested several strains in India, but we have not tested every. But we are very confident that CTS is doing their concerts with the other others that not exposed here because of some patent people. What we are seeing is what we have identified is compounds blocking the inferior. And influencer uses any transducer material. That is definitely faster and technical that. So still these pathways are to be covered by the setback. So we are expecting that typically by do very our compounds can be effective. That's what we call doing the COVID. So usually we start from either opposite. This is what the what's we tested this compounds were very delta. When we tested this compound, and we sent out paper, we will find that, and then we sent out paper. And then, while the paper was being reviewed, the Omicron stage. Then we are asked for years to test against Omicron, because Omicron strains, and this compounds were as so that is saying that it is one a single pathway is supported by multiple strains, the same virus or multiple viruses belonging to that. So therefore, we are thinking that we are not testing, cannot into the concept, but we can only expect that any string of right is good. This is one fine section of the work that is generally try to understand the kind of the whole factors. What's the sharing of the very. So this is one part that we think of the very good.
Is there any chance of the virus developing resistance against the compounds?
Sorry,
Is there any chance of the virus developing resistance against the compounds? Sure.
America,
Ah,
America, Institute and not really.
so is there a chance of the viruses developing resistance against this new compound?
Good question that we already tested. We tested the chemical virus evolution. The good thing is that since it is not targeting any viral protein, as we see for coming to targeting the viral behaviors. And the virus is already showed already should be like the for resistance. That's coming but our compounds of viruses so far has not showed any sign of resistance. The major reason our compounds are not targeting viral proteins. This compounds are targeting the cellular basis. The good thing is that there's no toxicity. Even if we shut down certain of the pathways, all our pathways are active. So there are multiple goals on the cell surface. At a given point in time, a couple of doors are open, but others are closed. And viruses use Some of these rules, we cannot use all the influenza uses in Latin, we can use that as one word, other like viruses, and there could be some other another goal. I think the first one there are two ways. One is depending on the presence of one device, one person starts to do either uses that password or you can take the step via history. In the endocytic group, it is visually captured mediated services, but here is showed that you can also pick up about that is different one. But what is converging here? All the pathways are converging with silicon lead. As I told you, the lead endosome is acidic here. And these compounds are blocking the acidification of lead. So if the viruses are coming from different roots, they are all moving to the endosome, and this is where the virus is. So the compounds are saying God spectrometry also constitutes the virus and it causes the disease. The program experience, this is also very much important that ICMR was very much interested uh, our study this short this because this is extremely important for pandemic stack document we also discussing. There's always the chances of another pandemic, and this is this is going to come. I don't know if it is coming in month or in coming 50 years or in our lifetime, but pandemics are taken to come. Either the influenza pandemic or SARS-CoV pandemic or some other new viruses. Because the viruses having their hosts, their circulating, in case the viruses influence significantly a number of mutations, we can see over flower species, as I said already, bombs or considers is serve as an intermediate host system and the immune. So, but the problem is, if we keep on targeting the virus, we will not be able to tackle pandemics. Uh, because case of new virus won't come out to one. So we have to come up with a strategy which will target the simple process, which is what by the virus or human. This is what we're trying to understand. Like this is one part of the work, and the other part we are trying to understand the first repertoire, which are helping the virus that is one of the most machineries that are described. And we have identified our own genes, we call them patent genes. And present with us, but somehow they're overwhelmed by the virus, the virus is infused, the virus is coming up with several alien strategies. For example, one example, the virus is using ubiquity, which many of you may know that it's a tag, it's one to attack what ubiquity, right? And the cell thinks that the virus is a chunk of protein that is received. And the cell employs what? Cell employs the motor proteins, which touch the viral proteins, and then there's a physical force which is generated, and virus is exploiting this to break open its capsid to release it. This is very delicate. Bacteria virus, for example, it kills the apple for minutes as it puts an apple cell. It releases bits, and that's why the virus. So all the viruses that involve elegant strategies in the first revolution, we get the cells. So now if we understand the first regime, how the host one for responding environment, then we can target host system. So we can target a set of host molecules and target the combination so that in case of any future pandemic breakout, we are equipped the recovery of drugs. So that we can prevent the casualties. This time we are not, but some people from our lab or labs, we try to hang together to be able to prevent. Will not be able to prevent the pandemic, but we might be that is what our opinion is. Suppose this scientific vaccination, that's another part spread here. But even after vaccination, we can say that we are infected. Right? Even after vaccination, we have died. Why? Because of the first our first is having from us our own genes and all. This is called pattern of forgiveness, and this is called mirror if you're in process.
Thank you for the inclusive explaining answer my question. If you have any other questions, just check on the if there are any questions. There are no further questions, then thank you very much once again. Nice to know how to deliberation. Not good here. You know, the problem teacher.
Thank you very much. Thank you, organizers, the problem program, all the we were expecting my message to our students that we're always excited about science. So many things, when I studied my PhD, my supervisor, this was not very my supervisor told me that like I need a very sport to find out like how the virus in itself. And I was super excited. I said, okay, I got a very simple topic. If I can understand how the virus in itself, I'll have my PhD. And then I looked up like what is existing because at that time I remember about 62,000 people from House Eastern. And I was happy and sad at the same time. Happy means like I thought that I got a very simple problem. And sad at the same time, thought that there are already like 62,000 papers in the conference. So that what more can I contribute to this field? But now I realize that 2023, well, that after really viruses for a longer time, that's we probably understand. We can see or even most of the stuff is still thinking we do not know. Have the number of things. Even for a very simple biological system like a virus, you don't understand the virus itself. You don't understand how the virus how all the force is involved. You don't understand how what the strategies the virus evolved over years. You overcome the possibility. Remember, when the virus enters the cell or in its body, it's a very extremely hostile environment. Right? It's very vulnerable antibodies, cytokines, blood flow, the immune cells, and still the virus animals is and humans. It's an enigma. And we are there, so it tries to increase. Thank you very much.
Thank you. Look forward to more questions in the future. So let's bring us to the any other similar program today. Those of you who have registered, so we will send you an email, feedback link. If you want feedback, you will read, please send it to us. Those are the program following now. Okay, very good. Now, before formally closing everything, thank you, all of friends.
Good afternoon, respected dignitaries, esteemed speakers, and attendants. On behalf of organizing committee, I extend my gratitude to everyone for invaluable contributions and active participation in the seminar from pathogens to pandemics on this World Zoonoses Day. My sincere thanks to our principal, Madam Dr. Shagata Dash Mahantaman, for her unwavering support in every aspect. I am thankful to our IQAC coordinator, Dr. Tapuri Jana Madam, that shown to our senior professor, Dr. Mara Bosch Madam, for joining us today. Please add a prayer to the night. I extend my gratitude to our guest speaker, Dr. Oliver Mitchell, for his interactive lecture on tracking zoonoses and potential pandemics in the 21st century. I extend my appreciation to our next speaker, Dr. Induni Banerjee, for his inspiring lecture, which enriched us on the topic. My heartfelt thanks to our technical support team members, Mr. Pablo Roy and Mr. Anil Mr. for continuous effort on your YouTube streaming that made the events run smooth. Thank you, our coordinators, Dr. Aniruddha Mitra and Dr. Kaveri. My sincere thanks to the professors, organizing committee members of our department, Shoma, Shubhamoy, Varnati Shambhamitra, Priyadarshini, Suranjana, and our lab attendants, Mithun, Anupam, and Pinku, for your continuous help. And last but not least, I would like to thank my beloved students. Your presence is highly appreciated and made this event successful. Thank you very much. Have a great day. Thank you.